Good day, Junior Techies, and welcome to this video where we're looking at bonus exam question 6. This is a question added to the series of videos just to help you prepare for your final exams. To practice these questions in applying exam technique is going to be our recipe for success. So let's just dive right in here. So we don't get much context to this question, but we can notice something very unique about these cells. We can see that there are 4 of them. We've got 2 unreplicated chromosomes in each of these cells, and they are bound by a nuclear membrane. Now, it says, study the diagram of a phase during meiosis below. So now the context said that these 4 cells are part of meiosis. If you think for yourself very, very quickly and what you know about meiosis, that there's only one phase during meiosis where we end up with 4 cells, and that is at the end of mitosis 2, which is telophase 2. So simply looking at this diagram and getting a bit of context from the question does help, okay, in analyzing exactly what those possible questions could be that are going to come out of this question. So let's see. Here, crossing over has taken place, so this is just an affirmation for us that this is in fact meiosis, and we are looking at telophase 2. Right, there are our 4 cells, and let's have a look at the questions. Let's identify the phase. Okay, we did that in gaining our context, so we can say that, right, we are definitely looking at telophase 2. Question 6.2 asks us to give 2 visible reasons for our answer. And here it says 5.1. So I'm just going to apologize for that now. And that is 6.1. So I'm just going to correct that very, very quickly there in the question. And let's move on. So it says give 2 visible reasons for your answer to question 6.1. And we can see here that if we just have a look at those cells again, we see that 1, there are 4 cells, which shows the reason for telophase 2, and each is with an unreplicated chromosomes inside the nucleus. Right, so this is just another bit of evidence which would support then the fact that this is in fact telophase 2. Question 6.3, how many chromosomes A are present in each cell in the diagram? Now that's pretty straightforward. But it does require of us to go to the diagram and actually count them. So that's how we do earn that 1 mark there. So we do have 1 mark for writing down 2. And now, how many chromosomes were present in the original cell at the start of meiosis? So again, this is testing our understanding of reduction division. So meiosis starts with 2n number in a nucleus, 2n number of chromosomes, and then at the end of the process of meiosis we have n number. So here each of these gametes at the end of telophase 2 have n number of chromosomes, and so my question asking me for how many were present in the original cell at the start, that would be 2n. So if there are 2 present at telophase 2, there would have been 4 present in that original cell. Or we can add to our answer here and say 2 pairs, because that's what we would have seen in that original cell. Now, question 6.4. This is a nice one. It addresses a little bit of genetics as well as meiosis. We're asked to name 2 processes during meiosis that lead to the cells being different from one another. So each of these gametes that arose from that one original cell are now genetically unique. They are not the same. What 2 processes during meiosis led to this? Okay, so there are a couple of things. We cannot mention anything outside of meiosis here, and there are 2 processes that you would have studied up on while preparing for your exam. And the first one there we look at is the crossing over of our homologous chromosomes, and that's in prophase 1, and then the random arrangement of chromosomes on that equatorial line during meiosis 1 and meiosis 2. So that's for 2 marks. So hopefully we got those written down correctly. And please just remember we refer to random arrangement of these chromosomes. 6.4b, explain the significance to a species of the cells being different from one another. So if these are gametes and each of the gametes being different from each other, or in terms of gametes being unique, it means after fertilization I'm going to end up with a increased genetic diversity in my offspring. Now that is a very, very important point to make mention of, because that's going to increase variation between the individuals that are born, so in those offspring, and that gives the species. Now we must remember the question asks us for the significance to a species, so we have to in our answer refer to species. This is going to increase the chance of survival of the species, because it would be able to endure whatever the environment or whatever environmental changes may be impacting on the species' ability to survive. So with genetic diversity, we have a greater chance that some individuals in that species will be able to survive the changes that they are being exposed to. All right, Junior Tuckies, thanks for joining me. This was a very nice question, not too long, good 10-mark question. I do hope that you found it interesting in the way that we're approaching these questions, and I do hope that it has helped you in your own method and exam technique in better understanding how to answer the question firstly, and secondly, how to know exactly what to write down to make sure that you maximize the number of marks that you're getting at the end of the day. Right, do join me for future videos. My name is Mr. P. Great for having you here.